All right, everybody, let's get started. Hi, folks. Welcome to the Lunchtime Discovery Series. It's good to be with you again for another edition of our program. In fact, the second episode of the Lunchtime Discovery Series this season. We just started back this program last Wednesday. Very exciting talk. Go check out the museum's YouTube channel. Visit the Lunchtime Discovery playlist. You can go see our chat with Preston Montague from Dix Park last week. Awesome stuff. This week, we've got another fantastic show, so I'm glad that you're with us. My name is Chris. I work here at the North Carolina Museum of Natural Sciences, and the Lunchtime Discovery Series is brought to you by us here at the museum. We host it, we produce it, but it's coordinated and organized by the folks in the North Carolina Office of Environmental Education within the Department of Environmental Quality. They do all the important behind-the-scenes legwork that brings this show together every single week. Make sure that you check out their office's website at eenorthcarolina.org. There you can see lots of programs, activities, and events just like this one, especially if you're somebody who's interested in environmental education, science and nature in North Carolina. Stay clued into what they've got going on because it's amazing stuff all the time. And the museum too. We do fun and interesting things too. So don't miss out on all the programs we've got here. Naturalsciences.org is the museum's website. In one of those two places, you can also see future Lunchtime Discovery Series programs. And at the Environmental Education Office's website, you can sign up for their email listserv. You'll get the email with this YouTube link in it to come and join us every Wednesday at noon. We always meet interesting people who do interesting work out there in the worlds of science, nature, conservation, education, and more. And we always have fantastic conversations. And I say conversation because that's really what we try to do here. We're going to take in a presentation, a talk, a slideshow from today's guest speaker. But after that, you all have the opportunity to voice your comments and questions into the program. All you got to do is jump into the chat on YouTube, type in your questions for our guest speaker and about today's topics. And after the talk, I'll read those into the program so that we can all be a part of this great big conversation on science and nature. And today's topic is one that I'm very excited about because we have one of the best science communicators in North Carolina. I think so. Everybody thinks so. It's unequivocal. Dr. Carly Ann York is with us today. Dr. York is a professor at Lenore Ryan University in biology, is a prolific science communicator and writer. You can find Dr. York on social media talking about all kinds of great science things. But today we're going to be talking about Dr. York's latest book, The Salmon Cannon and the Levitating Frog, which if that has not already captured your attention, I do not know what would. And Dr. York joins me now. Hi, Carly.
Hi, Chris. Thank you so much for that glowing introduction. You're very, very kind.
Well, I got started on this recently, pretty recently, and so I've only made it a few dozen pages in because I picked it up and I didn't have a lot of time. But it's really tough to put down. This is a great book.
Thank you. Thank you for saying that.
And your writing style is amazing. Also, like these stories of like quote unquote silly science are enthralling.
I'm so glad. It was really, really fun to write this book, and I learned a whole lot while I was doing it too. So, yeah, I'm excited to share.
We'll get into it then. I'll turn the program over to you.
All right. Thank you. I am going to go ahead and share my screen and get us loaded up. All right. So, like Chris said, I am going to talk today about my newest book, The Salmon Cannon and the Levitating Frog. So my goal in writing this book was to talk about some science that has maybe been considered a little bit silly in the past and talk about its real life value. So just a little more about me to start with. I'm an animal physiologist, which means I study how animals' bodies interact within their environments, how they've evolved to be in specific places. I think I probably have the best job in the entire world because when I'm not playing with animals. I get to be talking about animals with my students, and there's absolutely nothing I would rather be doing. So, as an animal physiologist and a scientist, my research has been a bit all over the place. I have studied equine social behavior. I have studied the sensory physiology of squid and how they respond to predators. And I have studied how African clawed frogs catch their food. So, my research has been pretty variable, and it's definitely the kind of research that some people might consider to be a little bit silly. So the way that I start this book out is a story about when I was working on my PhD at Old Dominion University in Virginia. At the time, I was also volunteering at the Virginia Zoo, and it was just a really nice thing for my soul to be around animals while I was getting through graduate school. And we would always go out to lunch with other volunteers after the morning chores were done. Um, and there was this one day I was having this conversation with another volunteer. He was an older, an older gentleman, retired veteran, pretty openly conservative politically. And he was asking me about my research. So at the time, I was studying squid, and I told him that I was studying squid biomechanics and kinematics, which are words that only scientists use. They don't really mean anything to anybody else. And he asked me why this was important. Um, and it was the first time that I'd really been asked why my research mattered, like in the big picture of things. And I didn't have a great answer for him. Uh, I mumbled something about like understanding the world from the perspective of a squid or something like that, but it wasn't good. I did not do a good job actually answering his question about why my research mattered. Um, and that has haunted me a little bit. I have since done a lot of work on how I talk about my research with non-scientists, but that was a moment that that really stuck with me for a while. And unfortunately, I'm not alone in being a scientist who um, has some trouble talking to the general public about what we do. As scientists, we spend years learning the language and the discipline of science, but we're never taught how to then take what we know and translate that back into something that non scientists can understand. And this has caused a lot of problems and certainly been a part of many of the issues that we're seeing today. Certainly with the mistrust of scientists and the spread of misinformation as well. So, one of the things that happens when we as scientists aren't very good at communicating to the public, what happens is other people start to tell our stories, and oftentimes it's politicians or the media, and they don't always get it right. So this is a quote from Vice Presidential candidate Sarah Palin. This one was pretty famous. It got a lot of attention when it was said, but tax dollars go to projects that have little or nothing to do with the public good. Things like fruit fly, research in Paris, I kid you not. So there are things about that um, are not correct. There are also some things about that are kind of correct. Um, so for one, the part that's not correct is like any disparaging remarks about fruit flies. Fruit flies are amazing. They are incredible model organisms. Pretty much like everything we know about genetics stems from fruit fly research. We've had 6 Nobel Prizes that have been won doing research on these flies. There's no limit to what we can study using them as a model organism. In fact, there's actually been some studies even on autism using them as a model organism, which Sarah Palin had actually said in the exact same speech that her nephew had autism. So it's too bad that she didn't know a little bit more about the kind of research that can go on with these critters. But one thing she was right about was that your taxpayer dollars do go to funding research like this. The National Science Foundation is the government organization that is the biggest funder of curiosity driven research in this country. So, the National Science Foundation was created in 1950, and it was created after there was a successful partnership between civilian scientists and the military during World War II. The goal of the National Science Foundation was to keep that partnership successful, supported, and moving forward, even in peacetime. So these were the major goals. We wanted to promote the progress of science, advance national health, prosperity, and welfare, and secure the national defense. So these are all positive things. I think we can all agree that these are things that we want to have for our country. Sarah Palin is not the only politician to have had questions about how the National Science Foundation spends taxpayer dollars. Um, in 1975, William Proxmire, who was a Democrat from Wisconsin, he started the Golden Fleece Awards. And these awards were given to things that he felt were wastes of federal funding. It wasn't just science. He was looking for wastes everywhere, but a lot of it ended up going to National Science Foundation types of research. And what he would do is he would take this and twist it to make it sound extra silly. This was never good news for the researcher who won the award. And times it was even extreme consequences like losing their funding because people no longer felt that their research was worth sponsoring anymore. When Proxmire retired, the torch was taken up by other senators. The biggest ones that did this were Jeff Flake and Tom Coburn, and they created these waste books where they mock again science mostly that has been funded by the National Science Foundation and therefore your tax dollars. Same kind of a situation where they take this and they twist it around and make it seem pretty farcical. Some of you all might remember about a decade ago or so, this video of a shrimp on a treadmill went super duper viral. This was everywhere and it got picked up by a lot of news outlets as well. And people were very, very angry to find out um, that this researcher, his name's David Shulnick, was given a $1.3 million NSF grant. And this was what was happening with that money. At least that's what the media wanted to tell you. Um, so I mean, Forbes advertised them as having a $3 million grant, which was just inaccurate. The AARP said that because of this research, senior citizens weren't getting the health services that they needed. It was even insinuated that like military spending was limited because the government was spending money on shrimp on a treadmill. So there was a whole lot of very, very negative press that went on surrounding this video and poor Dr. Shulnick's research. So before I go further, I want to just take a second and talk about research in general. There are different kinds of research, and the research that I'm talking about is basic research. If we were to say look at research like it was a tree, basic research is the root system. So this is curiosity driven research. The only goal is to gain knowledge. We are not trying to solve a problem. We're not trying to create a product. We just want to gain knowledge. So this is going to be the stability system for research as a whole. It's also underground, so it's not seen as much by the public, but this is incredibly important research to be done. On the other end is applied research, which is also incredibly important. This is the research that gets publicized a whole lot more, so we'll call it the visible trees and leaves here. This research is application driven, meaning you're going in to solve a problem, you're going in to create a product of some kind. Um, and that's what you're doing. It's a more narrow vision of how you're going to do your research. Both of these are really important. Um, Applied research doesn't have much trouble finding funding from a variety of sources, the government, but also from industry as well, because who doesn't want research that's going to solve products and or create products and solve problems versus basic research where you don't have necessarily something at the end of that, except knowledge, which may or might not be useful in the future. So the NSF was created really based around basic research. So Vannevar Bush was the major player in the creation of the National Science Foundation. He served as the science advisor to Roosevelt during World War II and helped get that solid relationship between civilian scientists and the military. And then he served as an advisor to Truman in the development of the National Science Foundation. So he wrote this proposal called the New Frontier, proposing what he wanted this foundation to look like. And in that, he said basic research is the pacemaker of technological progress. So Bush understood that we need to support this basic research in order for us to have a stable science innovation going on in this country. He knew that this was going to be a really important key, and it might not get money because there is no product at the end of it. But this is a public good, and so the government should be supporting this kind of research. And he's very accurate in saying that. Here's just two little stats to show his point here. About half of all economic growth in the U.S. can be traced back to some basic curiosity-driven research. This includes things like microprocessors and pretty much any technology that we're using at all today came first from some kind of basic research. At the same time, there was a study that was done looking at the origin of commonly used drugs. And what they found was that about 80% of them came from some basic research discovery that had no intention of ever creating a drug. So this work is being done without the intent of ever creating something specific, but having that basic knowledge, it pays off grandly in the long run. So these National Science Foundation grants, they they get a lot of attention, but they don't get a lot of attention in exactly how they're done. So David Shulnick wasn't just handed $1.3 million to put a shrimp on a treadmill. So he first had to write an incredibly intensive proposal. This proposal includes writing up the value of his research, and it also includes a very specific budget, how he's going to spend the money that he is asking for. Um, Once he submitted that grant, it then goes to a panel of experts who are going to evaluate this proposal for its merit, and they're going to decide whether or not to fund this research. It is an incredibly competitive process to get an NSF grant. The funding rates sit at around 25%, depending on the year that you're looking at. So that's tricky. That's going to be thousands of grants that actually get rejected. So if someone receives this NSF grant, you can be sure that the best in the field have decided that this is actually really good research to be funding. The other thing that gets a lot of confusion is how that money gets spent. So $1.3 million sounds like a whole lot of money, and it is a lot of money. Uh, but it gets broken down into ways you might not expect. So, first of all, um, a half or so of funds are going to go right into a bucket called indirect costs. This is money that goes straight to the university, and it's basically just money to keep the lights on, to keep the foundation stable, to keep the buildings rolling. This amount varies by institution. It can be anywhere from like 20% to even 80%, but it's a really big chunk of money that immediately comes out of that researcher's budget and goes straight to the university. The other thing that is usually not considered is the amount of money it takes to support and to train young scientists. Usually, a big chunk of that NSF grant is going to go towards supporting graduate students and postdoctoral researchers. And that'll be including stipends and salary, including insurance and tuition, um, depending on the institution where this is happening. It might look a little bit different, but it ends up being a good chunk of money that goes towards training and supporting our young scientists. Um, then we have the things that you might actually expect, like supplies and equipment. You do need to often buy or maintain equipment in order to do the most cutting edge research. You're going to have some fees, for example, publication fees. If you publish a scientific paper and you want it to be available to anybody who wants to read it, it's a pretty hefty fee to get that open access label. It's usually in the thousands of dollars. And then you have travel. It might be travel for research. It might be um, it might be travel for research. It might be travel to conferences, wherever you're going to go in order to promote this science. So those are relatively small slivers, though, in the big picture thing. The vast majority of an NSF grant is going towards the university and then towards supporting the training of our young scientists. So when David Shulnick responded to all the criticism that he was getting about his shrimp on a treadmill, he clarified a few things. So first of all, he didn't just put a shrimp on a treadmill for funsies. He was testing to see how the physiology of a shrimp changes when it is in polluted and like bacteria laden waters. So this is not only important ecologically, it's also important for shrimp fisheries. There's a lot to be gained from research like this. And at the same time, the money that he spent making this treadmill was less than $50. So that $1.3 million went to all kinds of other spaces. This was a little $50 treadmill. So, what I would like to do is share a few of my favorite stories from the book that started with just some kind of a curiosity driven research project with no intent of landing with some actual product or solution to a problem. Um, each of these started with just a curious researcher. So, this first one we're looking at, these are glowing jellyfish, and this is not AI. This is actually what these jellyfish look like. They glow, and there was a researcher who was really curious to figure out how they were able to glow. Um, we have a lot of different bioluminescent animals around us in the world, but he was specifically interested in how these jellyfish glow. So he isolated the protein that allows them to glow. It's called the green fluorescent protein. And this has actually been an incredibly useful tool in studying biological development and also in medicine. It took decades from the time this one researcher was studying how these jellyfish glow to get to the point where it could be inserted into different animals to track different cellular growth, to track nervous systems. And it took three different incredibly accomplished scientists to get us here too. So this was Nobel Prize winning research. It's obviously now beyond green. We have several different colors that can do this, but this has been a really, really big medical advance to be able to actually light up parts of the body. I'm sure many of you right now are familiar with somebody who is taking a GLP-1. This has been a really incredible medicine that actually goes all the way back to studying Gila monster venom. This is a Gila monster, and they are lizards that are found in the Southwest, and they are indeed venomous. But they also have a really interesting ecology in that they will bury themselves and they will not eat for months at a time. So the venom was interesting, but so was the fact that they could go for long periods of time without having to eat. So researchers were interested in looking at the protein composition of this venom. And what they found was that it was really good at stabilizing insulin levels, keeping blood sugar levels really solid. And this ended up being a really great tool for use in controlling diabetes. So Ozempic and other drugs like that were originally created for diabetes, and now are finding a really great use in helping people lose weight as well. But this all just started with studying some Gila monster venom. There were some researchers up at Duke University around the corner from you all. They were studying growth factors in baby rats, and they were having a really hard time getting their project to work. What they needed to do to study the growth factors was to remove the baby from its mom. And like many moms out there, mama rats were not thrilled about this happening. So they had to remove the babies for a longer period of time in order to actually run their experiments. But what they found was that the babies were not developing in the way that they should. Their growth factors were plateauing, and they were trying really hard to figure out why. So they ended up totally switching their project. Instead of studying these growth factors, they wanted to figure out why they weren't actually developing the way that they should. And they tested all kinds of different variables like pheromones and temperature and food and all the things. And what they ultimately found was that it was the mother's touch. So they developed a rat massage technique, and it is actually like a specific technique that you get trained in. It's not just any little rub down. And these researchers ended up meeting up with a psychologist who had been working with premature babies. And the way that hospitals used to deal with premature babies was that they would be completely isolated and fed intravenously. Um, but they also were not developing at the rate that was expected. So they took this baby rat massage technique, started applying it to premature children, and what they found was that it worked in the same way. So like their weight just like shot up, their heart rate increased, and on average they were able to shorten their hospital stays by like a week. So that's huge. That's huge for premature babies, for the parents of them, for hospital bills as well. And this all just kind of started by giving baby rats a massage. I'm sure a lot of you too have come across a cockroach at some point in time in your life, and maybe you tried to squish it. Maybe or maybe not, you were successful at being able to squish it. There was a researcher who was really interested in studying the squishability of cockroaches because they can be really hard to squish, and they can also fit through the tiniest little crevices as well. So he wanted to know like how they are able to do this and survive and tell about it. Um, what he ended up finding was that the shell on the back of that cockroach, it's not just like one solid piece. It actually has joints in it. And that's what allows it to squish down and bounce right back up. So he found that cockroaches can withstand like 900 times their body weight of being squished and come out completely unharmed. Um, and what he did with that was he created a little mini robot. So this is a roach robot where he took the biomechanic ideas he got from the cockroach itself and created this robot. And the way that this robot is being used now is actually in search and rescue kinds of missions because it can squeeze through tiny little cracks and it can withstand things falling on it. It's really, really durable and it can get to places that would be way too dangerous for people to actually get to. So, certainly not the product that he thought might come out of studying the squishability of cockroaches, but very cool technology that did. Another interesting story here. It's about how llamas helped with the COVID vaccinations. Anything with llama, I think, is pretty funny, anyways, because they're just funny. But this all started with a group of medical students who they were evaluating the composition of camel blood, and they found that the camel blood had these different kinds of little antibodies than what we have. And they're very, very tiny. They're called nanobodies. And through decades of research, that ended up shifting to llamas instead of camels, because apparently camels are really hard to keep. Um, what they found is that you can actually create a vaccination for people using these tiny little nanobodies. And these nanobodies can fit onto a virus and prevent it from actually attaching to a cell in a way that our own can't. So, for a while, this was actually one of the leading hopes for the COVID vaccine. This handsome fellow is Winter, the llama, who played a really big role in this research itself. This wasn't actually the research that ended up creating the vaccine, but it was a front runner for a while. And this is there's a booming biomed business around this called Llama Pharma, where we're studying all the different ways that these nanobodies could potentially help with medical issues. And this is the last one. This is my very favorite story that I wrote in the book, actually. Um, these are hero rats, and that's their real names. They are African pouched rats, and they're pretty big. They can be about 3 feet long when you include their tail. Um, and they have an incredibly good sense of smell. Like you can train them to discriminate between like so many different kinds of scents. And that research has been used to actually train them to do all kinds of different, very useful tasks. Um, so, over on the left there, the rat that's in that glass container, he's actually sniffing for tuberculosis. Um, TB is still a huge problem. It's one of the leading causes of death still in Africa, and the technology is really expensive and hard to maintain. But what they've found is that these rats are incredibly accurate at sniffing out TB in these different samples. So they train them when they sniff some TB, they'll give it a little scratch. And of course, this has been tested and followed up with, and they're really, really accurate at detecting TB. So a much less expensive, faster way of getting results. Over on the right is a rat that is helping to detect landmines. Um, there's still a lot of active landmines in Africa. And the beauty of these rats is that they're big rats, but they're smaller than like a dog. And they are small enough that they can walk over landmine seeded land and not trigger the landmine. So they can actually go and sniff out the landmine, and then it'll tell its handler that it's smelled a landmine and they can disarm it. They do actually do like weekly weigh ins to make sure that they never get above the weight where they might actually trigger a landmine. And then down below are two of their newer programs. The rat in the little red vest. I'm pretty sure her name is Jane. I have adopted like 4 of these online and highly recommend it. They send really detailed newsletters about everything they're doing, and it's wonderful. So Jane down there, she was being trained to detect like illegal wildlife trade. And the way that she does this, she has that little vest, but when she smells something that shouldn't be there, that ball that she has her hands on, she tugs it. And that tells people that she found something that shouldn't be there. And the one over to the right is a rescue rat. And this one is being used in search and rescue missions. In fact, I just saw this week that they've redesigned their little camera backpacks to make them lighter. But they too are trained to sniff out a person, and the camera isn't on until they find a person, and they let them know by tugging on a little thing, and then the camera comes on, and then people can communicate through that little camera. So just like really incredible things happening just because people were interested in figuring out how well rats could smell. So I want to take this just back to the bigger picture of the National Science Foundation and funding this kind of research. In 2024, the NSF budget was $9.06 billion, and the funding rate was around 26%, which is really quite low. Still, that's a competitive grant to get. But if you've been paying attention to the news, you have probably seen that the NSF is one of the places that is getting heavily cut funding wise. The Department of Efficiency slashed a lot of money from there. At this point in time, there have been about 50% less NSF grants awarded than in previous years. And there are researchers out there that are losing their research, and they are losing fellowships, and graduate students that are also not getting fellowships that they've earned. So at this point in time, the proposal for the 2026 budget is a huge, huge cut. It's $3.9 billion. And that would drop the funding rate to about 7% if things stayed pretty stable in terms of submitting proposals. So that is incredibly low. We would be missing out on so many opportunities to increase our knowledge base with a less than 10% rate of funding of this kind of research. And again, this isn't research that is easily funded in other ways. So the NSF has been the biggest funder of this kind of research for the past 50 years. And this is going to be the biggest cut that we have seen since its initiation in 1950. So when I started writing this book, I had no idea how prescient it would be at the time of its release. It came out in June, just as we were starting to see really big NSF cuts happening. And I have done a lot of interviews over the summer about it. And people ask me, like, what do we do? Which is a huge question. Like, what do we do? And I don't have like the answer to this very, very complicated question. But I have thought about this on a lot of different levels, and there are things to be done. First of all, if this is something that you are passionate about, make sure you're calling your local representatives and letting them know that this is incredibly important science that's getting done, that it needs to be funded. You all, if you're here today, logged on during lunchtime, you are interested in science, you care about science. don't be shy in sharing that with your family, with your friends. We need people to be really loud about science. There's been plenty of protests that are around, and if that's the kind of thing that works for you, go out and participate. If not, just talking to friends and family about this kind of research is something. Um, and then lastly, I just hope that if you hear about some research and maybe it sounds a little bit silly at first, you will consider that it could actually be the spark to like the next biggest innovation that we have. So with that, I'm happy to take any questions that you have, and I
Well,
will
wherever
share.
you're at, everybody, I'll encourage you to give a great big round of applause for Dr. York. Carly, excellent presentation. Wow.
Thanks.
These stories of what I'll say this. It's amazing to have read some stories in the book and then hear you talk about some of the others that I haven't got to yet. And for someone like me, like just hearing about how good the sense of smell that a rat has, like I'm all in.
Yeah,
If that's where the story stopped, like I'm 100% here for how good rats can smell and how potent Gila monster venom is. But then when you add on that a year later, 10 years later, 35 years later, we found out that these discoveries contribute to such important and incredible discoveries. Like the modern world is built off of, why a baboon's butt is red. And, you know, I don't think that one's in the book, but
it's not, but it could be, I'm sure.
right, like there's got to be something out there. It's it's greatly moving stuff to know that the process of science has such a profound impact on the daily lives of so many people in nearly every part of the world.
Yeah, for sure. In ways that we don't calculate at all on a day-to-day basis.
So, one thing that I picked up on in some of these stories, I wonder if you can speak to it, is the way that basic research gets communicated to applied research.
Mhm.
So often it seems like sometimes these things are coincidence or serendipity. Two researchers happen to walk past one another at a conference, or someone that, a genetics researcher happens to take in a presentation on animal behavior, and
Mhm.
then they start to build bridges and build connections. But I wonder if you can elucidate, shine some light on how these things happen, and maybe there's a technique in there for improving those pipelines, but
Yeah,
give us a
yeah.
sense of that.
I think you're absolutely right that sometimes it is just serendipity, and there are stories in this book where it just people just were in the right places at the right time. Um, And other than that, like science is an incredibly collaborative field. Most of these stories they include a number of different types of scientists. Like it'll start with an ecologist and it'll end with a geneticist. Like it takes people who are viewing the world from all these different perspectives to sometimes put it all together. Um, and as we celebrate collaboration in the science world, we celebrate it amongst disciplines and we celebrate it internationally. Um, and and I think it's incredibly important that there remains support for these different types of sciences in different places of the world to still be able to connect and talk to each other and work together.
How do you think we are there ways to improve that, do you think? Are there ways, like scientists are a collaborative bunch.
Yeah.
This I've experienced, you know it well. Um,
So
it's
one thing that the NSF has done
yeah.
is it has actually rewarded collaboration like that. It has um, funded it. Part of what they look for when they are funding is the kind of collaboration that can happen within this project itself. So For example, like my squid research was funded by the National Science Foundation. We had a fluid mechanics engineer that was on my doctoral committee and on that grant as well. So while we had the biologist who was dealing with the squid and what that squid was doing, that data was then actually going to a fluid mechanist who was able to actually think about how that might work in terms of like robots or any other technology. So NSF has historically rewarded that kind of diversity on an NSF team. It would be great if it would continue to do that. It would be great if it would continue to fund anybody, please. But yeah, and also just yeah, another thing that would be lovely is if journals didn't have papers behind paywalls. That can be sometimes a really big roadblock when it comes to sharing ideas. And it's if you can't get access to the information, it's not gonna go as far as it possibly could. And you know, for a public good, I think it's incredibly important to share that. But right now, you do have to pay a considerable amount of money when you publish in order to have your papers be open access available for other scientists that aren't members of that journal and for the public as well. And I think it's incredibly important that the public has access to these journal articles as well. I doubt they would want to read them. They're pretty dry, but they should have the access for sure.
Yeah, absolutely. Thank you. Excellent, excellent answer. One from our viewers online. What are some ways we can show folks that this exploration is worthwhile and deserving of funding? Something really in your face.
Something really in your face. Uh, well, a lot of the examples in the book, um, that was kind of my goal, was to provide some examples that were pretty in your face, that were pretty weird and out there. So if you haven't read the book, I'd highly recommend it. And some of those stories might be exactly what you're looking for to maybe share this with the people around you about the value of this. There's also just some incredible statistics out there, like I just showed you those really brief two about the economic growth and medicines that come from this basic research. I have more of those also in the book that I can't quote right off the top of my head, but there is a solid amount of data that says like, no, this is like really valuable. This is really, it's worth it in the long run for us to fund just gaining knowledge, for us to fund just like having this solid base.
It's always interesting to me to talk about basic research and its value because there's always the polar view, or it's not it's not totally a polar view or an opposite view, and one that I've heard across scientific disciplines, right? Why should there be shrimps on treadmills? Why should there be rockets going to the moon? There are plenty of things going on with people on ground that need urgent and dramatic solutions. And it's really difficult, I feel like, to communicate the value of basic research. Like, I think your book has done just like a spectacular job, right? Because there's so many wonderful examples. I guess I still worry about the sense of urgency
Mhm.
and the need for solutions,
Mhm.
and
And that
and then how we learn to communicate the value of science that way.
it's always going to be a toughie because people want the instant gratification of finding the answers, and the reality is we haven't found the answer because we don't have the knowledge that we need
Mm.
to find the answer. And that could come in the weirdest of places. And it also might take 30 years before we're able to piece it all together. So it's not instant gratification. And I understand why people are like, yeah, but we need to do this now. Um, Ultimately, you're selling yourself short in the long run if you stop gaining knowledge. Like you You can only use what you know. And if we're already using what we know, then we don't know enough. We don't know what what the solution is to this particular problem. And we might find it in the mouth of a Gila monster. I don't know. It's just we just simply don't know. Um, also with a lot of these studies, when they are talked about in the media, they are twisted so much to sound so dumb, um, that I mean, and I know that's it's very purposeful to say that this is wasted money, but I would encourage people to do a little research on their own about this kind of stuff, because you might find really quickly that, like, oh, this is valuable in and of itself. Like studying how shrimp's physiology changes with bacteria laden water is actually really important, like right now. That's not something we even need to wait 30 years for to see an outcome for that. So it's tough. It really is. And it requires people to use a little bit of, um, to be patient and to use their imaginations a bit to realize that like this is worth it to put this money into this.
And even, you started off at the very beginning talking about the value of good communication,
Yeah.
about why we think some of these things are important, and the process that goes into them, so that folks don't necessarily have to use their imaginations even.
Right.
We can be sharing openly and in a transparent way, uh, so that folks can, even if they see something that they feel doesn't address a need that they see, we can understand that this is the process that gets us to answering big questions.
Yeah, sure. For sure.
Let me throw some more questions your way from the chat because they're rolling in on me.
Yeah.
Lisa wants to know if you have any plans to have a young adult version for teachers to use with younger students in the classroom.
I don't have a plan for that. But that would be pretty cool, wouldn't it?
Would be pretty cool.
I could think
And
about
we've
that.
got a high school physics teacher in the chat too, who's curious about demonstrating it in the classroom.
Yeah. Yeah, demonstrating it in the classroom, like with an experiment, is a little tricky because again, there is this like long haul with the
Mhm.
basic research stuff. I don't have um like an experiment example. Do you?
Get in touch with us here at the Museum of Natural Sciences because
Okay.
we have devoted some time and energy to activities that are for classroom learners on research pipelines. For example, we've got an activity that we used to use that was a board game about getting something like Gila monster venom into pharmaceuticals,
Oh, that's so cool.
like the pharmaceutical process and drug discovery and things like that. So uh, I feel like there's lots of answers to that, and we might
Okay.
have some of those here at the Museum of Natural Sciences.
That's awesome. I would like to hear about those as well.
We can chat about them because everybody, thank you for tuning into this program. If you would like to actually pick up a copy of The Salmon Cannon and the Levitating Frog, you can do that here in downtown Raleigh tomorrow night. At 7 o'clock here at the Museum of Natural Sciences, we'll be hosting the Science Cafe, and our very special guest in person will be Dr. Carly Ann York, who will be talking about the book again, yes, but we'll be doing the presentation in person and in the cafe, so you can grab a bite to eat, get something to drink, hang out with us. And we've got plenty of copies of the book that'll be available for sale. And Carly, you might autograph the books.
I will do so. Not well, but I will do so.
I think it would be so much fun. We've got a few more minutes. Let me throw another question
Sure.
at you. Because I'm curious about your research for pulling the book together. Because so many of the stories, right, we at this point they have the benefit of hindsight. But
Exactly.
how do you connect all of the dots on some of these things? How do you come to understand how these processes played out? Some of them I get, like, shrimp on a treadmill was this huge thing,
Mhm.
uh, lots of stories, and then clear ending.
Yeah.
But I sense that a lot of these things may have been difficult to piece together.
Yeah, they really were, and this is my first time taking a swing at research like this. Um, it was fun because you kind of just go down rabbit holes. Like I did not plan all of those stories that I told like in advance. I had an outline, I had places I wanted to go, but I would be researching one thing and it would lead me to another thing and then to another thing. And I would have a completely different story by the time I finished reading as much as I possibly could about it. But yeah, it is piecing it together. It's like looking for this person's story and this person who did this one little thing, go and look for their story. And one of the actual hardest ones was the COVID and the camel one. There was lots of information about the nanobodies, about using the llamas, but I was having a really hard time figuring out how they found those nanobodies. Like somebody somewhere first looked at this blood and said, that's weird. So I had to do a lot of digging to get back to that story. And I mean, it went all the way back to Belgium in the 1980s and this group of medical students. So it was worth the trip to go down and figure out what it was. But yeah, sometimes it did take some serious searching around to figure it out.
Were you just like combing through the references sections of papers trying to find the right titles and
Basically, yeah, like reading
gosh,
all the references um and then just doing my own little offshoots here and there too. So but it was actually really fun to do. I enjoyed it.
fascinating stuff? Well, Carly, thank you for joining us on the Lunchtime Discovery series today.
You are welcome.
Excellent presentation. Folks, the book is The Salmon Cannon and the Levitating Frog and Other Serious Discoveries of Silly Science. You can pick up a copy of the book tomorrow night here at the Museum of Natural Sciences at the Science Cafe. More information is about that at naturalsciences.org. And of course, you can follow Dr. York on social media and get share with us your website and your socials. That way people can keep up with what you've got going on.
Yeah, I am biology Carly on all the places pretty much. I'm still on Twitter. I'm on Blue Sky and Instagram and TikTok, and I'm biology Carly in all of those places. My website is a Wix website, and if you Google me, I'm sure it'll come up somewhere, but it's not like an elegant title.
Oh, you know what? We already put it in the chat.
Oh, great.
It's
Okay.
right there at the top of the chat. Everybody, you could scroll up,
Fantastic.
click, jump right in. Perfect.
Awesome.
All right, everybody. Have a great rest of your Wednesday. We'll see you again here next week for another edition of the Lunchtime Discovery series. Next week, we are talking to the Secretary of the Department of Environmental Quality, Reed Wilson. So don't miss out. We're going to have a great conversation about the environmental landscape in North Carolina with the Secretary of, of the Department of Environmental Quality. It's going to be great. We will be in person in the SECU Daily Planet Theater, so you can come and grab a seat here right in downtown Raleigh inside the Globe, or join us right here at the museum's YouTube channel again next Wednesday at noon. Take care, everybody. Have a great day. We'll see you soon.